Okay, thank you everyone for coming today. We really appreciate you making the trek outside in this lovely weather that we're having. So I won't take up too much of your time. I would just like to introduce our guest speaker, Dr. Lundy Pentz. He comes to us, he's a professor emeritus from Mary Baldwin College, and he's a graduate of Johns Hopkins University with a PhD. And he will be talking on cells, tissues, and money. And I will let him take it away.
Okay. Well, I figured I have suffered through enough interminable PowerPoint presentations. I wasn't going to inflict that on you. So I want to just talk through a couple of things about this book. Raises some very important issues. It's a very absorbing book. If you come to it as I do from a science background, there are also several things that make us go, ooh, but why didn't you tell them this part of the story or that part of the story? There are, in fact, some things that are a bit misleading in there. And so I thought it would be good to kind of focus on the money story. And to remind you, one of the contentions in the book is that when Gay developed this cell line in '51, ever since then, someone somewhere has been making money off the cells. Okay, that's on page 195 if you want to check me. And obviously, since the Lacks family, as the book richly illustrates, has not benefited in any way by this, there's been an injustice perpetrated. She also cites Invitrogen selling HeLa products for $100 to $1,000 a vial, and also American Type Culture Collection, which the book is a little out of date, $256 has gone up since then per vial for frozen HeLa cells, and says that since HeLa is, quote, one of the most popular cell lines in the world, the profit from HeLa cells is surely significant. Okay. Well, that's where I kind of go, wait a minute, that's not entirely fair, because what the book lacks, except for some really weird descriptions of part of what Gay had to do in '51 to get the cells into culture, doesn't really tell you anything about how tissue culture and cell culture are really done. It's also a bit timely and relevant because although, as you'll see, I'll kind of mention the idea that HeLa cells are wildly popular is a really old idea. Most of the people I know that do any kind of tissue culture have gone completely over to the side that they don't want HeLa cells anywhere near their labs. So this is really bad stuff, and we'll maybe talk about why that might be. But I thought, gee, this is really timely because just this year, as you probably know, Planned Parenthood has taken a lot of flack for selling fetal tissue for research. In fact, I just noticed a full-page ad that a bunch of medical researchers took out in the Washington Post begging Congress not to politicize this. Good luck. But that is foreshadowed in a way by Skloot's comment in the book that it's legal to sell human organs and tissues for transplants, or it's illegal, sorry, to sell human tissues and organs for transplants or medical treatments, but it's perfectly legal to give them away while charging fees for collecting and processing them. And while she doesn't exactly say this, I think you can hear the kind of, yeah, sure. So you just set the fees to whatever you want to make a profit, and it's just a way of getting around the law, right? So it's these unreasonable fees of several $100s for collecting and processing that are the real heart of what I wanted to talk about. Besides that, I've always had a little bias about this because I think you don't really understand something until you've tried to do it. And getting in there and trying to grow cells in the lab is something that I think everybody ought to be able to do, frankly. Of course, again, I'm biased. I went to Hopkins, and I was there in the '70s in the middle of the hair-raising, all of our cultures are contaminated with HeLa cells thing. For my own doctoral dissertation, part of what I worked with is a line of hydatidiform mole cells. This is a rare type of placental tumor. It's not fatal, no tragic outcomes in this case, but the cells were a great model for looking at why the mother tolerates a fetus for 9 months, when if it were any other foreign tissue, she would reject it in a month. So it's an important question to answer. But that was a human cell line derived at the Hopkins Hospital, and I worked a great deal with that, and along the way with dozens and dozens of other mouse and human and other cell lines. When I came to Mary Baldwin in 1980 to teach biology, I said, we've really got to get this fixed up, and we actually had a small lab that was used for tissue culture. So, I've managed to build it up a little bit and started in cell biology, having the students culture cells for their projects. And of course, senior research projects depended very heavily on cell culture. But in cell biology, my sophomores would start right out and say, okay, do you want to work with an established cell line like HeLa cells? We always had those around. Or do you want to try to create your own culture from a mouse? And particularly, sometimes it would be a normal tissue from a mouse, sometimes a tumor. And the students would go through all the steps you need to do to create a cell line and would get the experience of what it's like. Now, there's one wrinkle on all of this. So I've had a lot of experience with this kind of thing. There's one thing you never tell the students, at least I don't, because I don't like to scare students. All right, and yet as the person who would, back in the day, have to order all this stuff, I was keenly aware of how much everything cost. And so when a student would spill a vial of fetal bovine serum or something, I would go, oh, that's all right, don't worry, we'll clean it up. Let's see, are we going to be able to afford next week's reagents? And so I was very much aware of the cost of all of this. And that's, in a way, I want to talk you through briefly today a little bit of what do you have to do to get cells into culture from a tumor, and what does it cost to do it? And I hope I'll kind of make some light bulbs go off here. And then maybe, time permitting, we can go into some other things. But I'd really like to kind of, and I should have said, like to stop for questions early. Second thing is, please, for heaven's sakes, if I say something that doesn't make sense to you, or something occurs to you that you want to discuss, just interrupt, okay? There's nothing worse than sitting there smiling and nodding politely when you have no idea what in the heck is going on. So, well, let's suppose then you're one of my students. You come to me and you say, oh, I saw this mouse in our mouse colony. It has a bump on its back, and it's probably a sarcoma or some kind of skin tumor, and I'd like to try to put it into culture. Can I do that for my cell project? I was like, fine, okay. First thing you got to do is know about keeping things sterile, and we go over that a lot. The short version of it is, I wish I could do this. I never had the nerve to, but imagine that you would come in and we'd collect some kind of powdered dye, like fluorescein powder, that you could just stick your arms into up to the elbow, put your face down in and shake, get it all over your hair, and then walk in and try to pour things and pipette things from one container into another and never get a speck of that dye into them. That's what sterile technique is. If you've ever been in an operating room, you know there's a line on the floor. If you're not scrubbed, you don't cross that line. And you probably also know that when a surgeon has to do something involving getting in and doing fine work, they always wear a great big sterile shield. That is to keep their bacteria from dropping into the incision, okay? The one thing bacteria never learned to do, thank God, is fly. And so they can't jump or fly. They can only fall or wash from one place to another in liquid. So a rainy day, bacteria can spread, but you don't let it rain in your sterile work area. So what you got to know is, first of all, learn how to wash your hands properly and clean things up. And then, of course, you got to take the little mouse and soak him with alcohol, because his fur is all full of bacteria. Of course, you've already euthanized him. And soak his fur with alcohol. And then you carry him into your special roughly $20,000 to $50,000 BSL-2 cabinet. We used to use laminar flow hoods that blew sterile air across the work area and out to you. And of course, now that we know about all the viruses that cultured cells grow, we don't like to do that. So instead, we have a sterile curtain of air going down across the opening. So you're working in what looks like a brightly lit fume hood, just a cabinet, that you can put your hands through the curtain of air, and everything inside, if it was sterilized to begin with, will stay sterile, and you will be protected. And of course, that's the idea. So you put your little mouse in there, and then you've got to sterilely open him up. Well, that's the easy part. We've been sterilizing scalpels and forceps for surgery for 100 years. People know how to do this. You can actually just dip them in alcohol and light the alcohol and let it burn off, and that does a pretty good job. Problem is, don't do what one of my students did, which is to not let the flames go out before she goes to work on the alcohol-soaked mouse. Mouse flambe there. To make matters worse, she tried to put the fire out with a squirt bottle filled with alcohol. Very toasty mouse. So if you don't do that, then you can open up that skin and reveal the tumor, which is usually not too well attached. And now you need to know something about tumors. They're dying. They're dying from the inside. So you don't want that whole tumor. A lot of students, oh, let me take the whole thing out. No, you're not a cancer surgeon. You're just a cell culture person. You just want the fringes of the tumor. You want the parts that are still likely to be alive because they're not starved for oxygen. You know you're in trouble when a tumor has a good blood supply, big blood vessels you can see going into it. That's a bad thing. Then they're not dying in the middle. So you take the outer area, and you take out something about the size of a grain of rice from it, and with your sterilized instruments, and then you turn around and say, Dr. Pentz, I need the enzymes. Okay, and I come over to you with a little tube containing enzymes called trypsin and collagenase, which are really good at chewing up the proteins that stick tissues together. That is, that stick the cells together that compose tissues. And if you throw the tissue into those enzymes, in perhaps for a chunk of tissue like that, 15 minutes to 20 minutes with gentle shaking, the solution will get cloudy. And the cloudiness is the cells peeling off the surface. They had their adhesion proteins got chopped by the enzymes, and now they're drifting around as single cells, which is what you want. Now, I said I'd talk about cost. This isn't too bad. You see, these enzymes are pretty cheap, and they're available from microorganisms, so you can grow as much as you want. So we may say what I'm handing you is about $4 worth of stuff to dissolve your tumor into single cells. And now what are you going to do? You see, if you leave those cells in the enzymes for about 10 minutes more, you'll start killing them. I discovered a cure for cancer, right? Problem is, this would be equivalent to injecting meat tenderizer into your patient. It doesn't work too well when all your connective tissue falls apart. So we can't really use it as a cure, and we have to be aware of having a way to stop it. How do you stop the digestion? Give it something to chew on that isn't your cells. Namely, give it some growth medium. And here's the biggie. The growth medium is a mystery substance, and you realize what a medium is, right? You know, as in the medium is the message and all that. It's a stuff. It's a liquid or solid stuff that probably is involved with supporting or growing something. So, you know, people that are into horticulture grow their orchids in special media. This is the same thing. It's a liquid that is used for cell culture and was just being developed when George Gay was working, was perfected through the 50s and 60s by a variety of people, some of whom are worth reading up on. And I strongly suggest, if you want a woman in biology, look up Honor Fell, Dame Honor Fell, a dame of the British Empire, because she figured out how to get the first mammalian cells to grow in culture. And brilliant, brilliant woman. But the stuff they had to go through was amazing. So what have we got in growth medium for your cells and mine? And by the way, this applies equally to human and mouse. It makes no difference. You use the same tissue culture media on both, and any other mammal pretty much. Well, we need about 13 or 14 of the amino acids, not just the ones we call essential, because not all cells can make all of the amino acids that your whole body can. And we've just taken the cells out of the body, so we got to give them extra amino acids, lots of them, about 8 to 12 vitamins, fatty acids, sugars, the whole shopping list basically of all the nutritious stuff that's in your blood. And then we've got to add stuff to maintain the pH properly. You don't want this to become too acid. And so typically what we do is put a pinch of baking soda in bicarbonate, which makes it a bit alkaline, but we're going to put these cells in an incubator that has CO2 gas in it, and that will bring them back to a nice neutral pH and keep them there. So they'll be just as neutral as anything you'd ever put on your hair, and that's what they need. So that growth medium is concocted out of all these nutrients, some stuff to stabilize the pH, and one other thing. Serum. Well, serum is the liquid part that's left over after your blood clots. It happens naturally. If you take a blood sample, leave it in the refrigerator overnight, come back the next day, you'll have a little clot that's shaped like the inside of the vessel, but has peeled away from the walls. It's about half as big as the blood sample, and the other half is now a straw-colored liquid that's properly called blood serum. And they found out early on that serum works for some things, but not for everything. And really, if you want human or mouse or most other cells that are normal to grow, you've got to give them not any old serum, but a fetal serum. And fortunately, bovine serum, FBS, is what we use. Now, this is a rare substance, but now and then a cow that actually is carrying a calf will show up to be slaughtered in a slaughterhouse, and a few, not all, but some slaughterhouses know to take that fetal calf and bleed it and allow the blood to clot and collect the serum because they can sell it for big bucks. Okay, for a 500 mil, that is about a pint of this stuff, we're talking in the range of $600 to $900. Okay, so this is a big ticket item. So I'm just going to flag it here. And I'm going to give it the, just as a rough reminder, I've got a spreadsheet on all this, but you probably don't want to go through that level of detail. Unfortunately, you've got to make your medium about 10% in this very expensive stuff. So all of a sudden, to stop the enzymes from killing our cells, we got to put this really expensive stuff on there and let the enzymes chew on it and destroy it. And then we got to get the cells out of it, put them in a centrifuge and spin them so the cells come down to the bottom of the tube, pour off all that liquid from the top, and replace it with, fresh medium with fetal calf serum or fetal bovine serum in it. And so we just kind of did a double whammy on the cost, resuspend the cells in that, and then you put them, well, what do you put them into? Well, let's take a little look at this. What do you think of as the typical scientific thing? If you say he's going into his lab and he's going to work with test tubes and what else? Yeah, Petri dish. Julius Petri was one of Robert Koch's graduate students, and he designed a shallow dish that you could actually pour agar into and make a bacterial growth medium. There's a loose-fitting lid. Why loose? Well, cells need to breathe, right? Oxygen needs to be able to get in there. And these can be made of glass, Pyrex, and they can be sterilized if you have a roughly $30,000 autoclave, giant pressure cooker to sterilize them. And they're used to this day. But back in the 70s, we were still using them for tissue culture. And the other thing, I've been talking about pipettes, these long calibrated jobs in the old days. We still use these too. There's a modern version of them. And so many mils will be pipetted into your plate, so that all works. But right in the 70s, right as I was getting into this, they started coming out with a new thing called a T-flask. And this is kind of like, as I say, a cubist's idea of a bottle. A T-flask has a cap and threads and square sides. And the kind I'm talking about, we'll use consistently for this, great for student projects, is a little bitty thing called a T25. It's about 1 inch by 2 inches by 3 inches. And the cells are going to grow on the bottom of the flask when it's sitting down on its side. So we've gone from sterilizable glassware that you reuse to plastic. Can you sterilize plastic? Anybody know? Well, you don't really want to. There's two ways to do it. One is to expose it to such intense gamma radiation that nothing can survive. Okay, there's a device called a gammator that will do that for you. But of course, you have to be licensed by the Nuclear Regulatory Commission, and the thing weighs about 6 tons, and it's hazardous, and there aren't very many of them. The other way you can do is to put it in a gas chamber and infuse it with ethylene oxide gas, which is quite poisonous. And again, you just don't want to do that in your lab. So people's autoclaves mostly are rusting away now because what they have done is to go over to buying pre-sterilized plasticware that's single use only. Environmentalists take note. Okay. This has its drawbacks. But it does one thing, it keeps things sterile. We may have time to get back to how did everybody's tissue cultures get infected with HeLa cells? Reusable glassware. But I stray from my main point about money. So let's look at the money. What have we spent so far? Well, remember I told you how expensive the stuff is. We've just used up about $10 more growth medium. And now we have to put the cells in the tea flasks. And you don't put them in just one. You put them probably in about 3 or more. Because we have to replicate stuff in science, right? And besides, how do you know you didn't get your thumb into one of those flasks when you were working with it? Did I mention you don't use surgeon's gloves in these hoods? They make you very awkward unless they're really good-fitting gloves. So you just wash your hands with alcohol and keep your fingers out of things. That's my advice to people anyway. And then you've got to stick these things into a tissue culture incubator. Marvelous thing with a water jacket so it keeps a constant temperature, blood heat, 37 degrees. Has CO2 tanks hooked up to it. That's because your cells are sitting in a medium that has bicarbonate, which is alkaline, right? And that's stuff people used to tell you to put on acid burns, not a good idea. But when you get the CO2 gas, like any carbonated beverage, that makes it a little more acidic, so it balances. And you get a nice pH, and a pH indicator called phenol red in the medium will tell you that. You can see that the culture is bright red. A lot of people look at this and say, oh, you're growing them in blood. If your blood looks like that, you're in trouble. This looks like cherry Kool-Aid. And it has the virtue of when you go a couple of tenths low, it gets decidedly orange. And go a couple of tenths of a pH unit high, it looks quite purple. So you can't miss it when the pH isn't right. So that's an advantage.
If you see a color change, can you adjust it?
Yeah. But with mammalian cell culture, you don't. In other words, if the pH changes, if it goes purple, then something is really seriously wrong, like somebody actually put something into your culture that was very alkaline. What they'll usually do is they'll go a little bit yellow, a little bit on the orange side, and whoa, it's time to do the next thing. You see, Nicole is feeding into this. The next thing you have to do in a week or a few days, depending on the culture, when you see it start to turn a little orange, you got to give it fresh medium. So instead of correcting the old medium, you say, oh, the cells were excreting acid byproducts. If you look at the culture, which you can do, there are microscopes that look up at the bottom of the plate, you can say, oh, well, it's starting to grow. The plate's covered with cells, and I don't want them to poison themselves. So what you do is suction off the old medium and put on new, right? Now, what you're going to find out in, again, weeks probably, if you're lucky, is how fast can these cells really grow? Little caveat on the books, HeLa cells do not grow faster than any other cell ever cultured. All cells can grow relatively fast. The record for human cells is 16 hours to double, and that's for your normal lymphocytes. When you make an immune response, those cells really start dividing fast. That's why your lymph nodes swell up so rapidly. But HeLa cells grow well, but they don't grow any better than thousands and thousands of other cancer and normal cells. And what will happen is that if you ignore that warning sign, the little orangey bit, and you say, or you look at them and you say, oh yeah, they're starting to touch each other, get confluent as we call it. So they're a little crowded, but I don't feel like dealing with them today. I'm going to come in tomorrow. You come in tomorrow, and they're going to be peeling off the bottom of the plate like a bad sunburn, like your skin parting company with you, and they're done. It's over. Put them away. They're dead. So you really have to split your cells. Now, what are you splitting? Other operation. That is to take one plate of cells that were getting crowded and get them off the plate and put them into not one, but usually about 5 other plates. And you talk about the splitting ratio. And 1 to 5 other plates. Oh, wait a minute, what are you going to have to put in all of those 5 other plates? We're getting into expensive territory now. So the splitting requires enzymes like our first step. The enzymes aren't expensive. The serum, however, which you've got to put in there is. And essentially, here you are, you're stuck now, by the way. You've got flasks that are going to increase in number, 5 this week, and 5 times 5 or 25 the next week. You see where we're going with this, right? This is population growth. This is exponential growth. And so students are always excited, oh, look, my mouse cells are growing. And then the next week, there's my mouse cells are growing. Well, what do I do about this? I've got 25 plates. I can't keep up with all of this, right? So what was a joy becomes a problem really fast. And you're telling me about it because I'm sitting there writing the purchase orders for the stuff. Let's just say that we're feeding flasks, and feeding costs only about $3 a flask. How often do you have to do that? Every 2 to 3 days. Okay, so you can't take a long vacation when you're growing cells. 2 to 3 days, have got to be fed. Most cells will need to be split every 7 to 8 days. A lot of variation there depending on the cell type. But HeLa cells are no different, by the way. They need to be split about every week. Well, splitting costs about $25 for each flask split. But remember, you then, if you're going to grow them all, and you keep them going, in the beginning we went from 3 flasks. So because we had 3 to start with, that's not, 1 to 5 would give you 15 flasks. And at that point, you've grown about 150 million cells. And adding up the cost of all the medium, you've spent about $180 doing it. Now, sometimes you get a student who gets a little carried away and says, I want to keep growing them. This is so cool. I can grow all I want. And next week, your second split, 75 plates, 750 million cells, and about $600 spent. Carry out the third split the next week, 375 plates, 37 billion cells, bill of over $2,000. And hopefully, by the time you get to the 4th week, your professor says, hey, wait a minute, what are you going to do with all these cells, right? You're over the top. But you see how it adds up, right? Then this is talking about 1 cell line that we were just trying to start from 1 mouse. Now, what does that mean in practical? One thing, if you call up somebody and say, oh, are you growing cell X, like HeLa cells? If they are, do you think you can say, would you give me some?
They've got plenty.
Yeah, it's like people that make yogurt. You know, once you get started, you either have to stop or you have to get rid of the surplus. Right? Say, absolutely, and how many plates do you want? Take them off my hands, please. Right? And so file this away when we talk about things like American Type Culture Collection charging for a frozen vial of HeLa cells. Okay, any researcher who's working with the cells will happily give them to you. There's just one constraint I want to mention. But so the other thing you can do, of course, is freeze your cells. And freezing them works. Freezing them is great, but you can't put them in the freezer at home, right? You got to buy a -80 freezer. Home freezers are -20 Celsius, 21. A -80 is a lot colder, and that is only to get the cells down to -80. That isn't really very good for storing them for very long. To store them for a few years, you've got to freeze them in liquid nitrogen, -196 Celsius. Very cold liquid. And to do that, of course, you need special little freezing vials that are made of a hard plastic. And you've all seen this in movies, right? Like the original Jurassic Park. I love it, where Nedry goes in to steal the dinosaur embryos, and he yanks the cane right out of the freezer, and there's no water vapor condensing, there's no ice crystals forming, and he grabs the things out with his bare hand. You try that sometime. You won't let go. It freezes to your fingers. Very unpleasant. So anyway, you can freeze your cells. Now, here's another one. You may have become aware of this if you were paying attention a few years ago to the whole stem cell debate. Can you leave cells in liquid nitrogen forever? You see, gamma rays are still going through that freezer. The DNA of the cells is still taking all kinds of hits, but what you did stop is any significant enzyme activity to repair the genetic damage. So the longer you leave your cells in that liquid nitrogen, the sicker they are, the less likely they are to recover when you try to thaw them. And the thawing, by the way, is when the killing occurs. When they try to thaw out and try to transcribe their damaged DNA, a lot of them don't do so well. By the way, also, don't ever pay anybody to freeze your body in liquid nitrogen so you can be brought back in the future, as people do. There's people that cheaper to freeze your head only, but some people want the whole body for some reason. The thawing is when the mischief occurs. Well, so going back to the economics here, what do you have to do, even though you realize this is temporary? So splitting a flask of cells is about like feeding them, $25 or so. Not a big deal. Problem is when you go to freeze them, you need a cryoprotectant, long word for a small molecule that gets into the cell membranes and prevents them from becoming damaged by freezing. And we use dimethyl sulfoxide as solvent, DMSO, which unfortunately costs about $5 for a milliliter of tissue culture grade. So again, this makes the cost of freezing cells ascend quite remarkably. And then you're going to put them in liquid nitrogen, right? Now, we have an advantage here in Virginia because we have a lot of cattle farms. And that means it's easy to get liquid nitrogen delivered because bull sperm is frozen in liquid nitrogen. And no cattle breeder in his right mind has a live bull on his property. He just orders the sperm and keeps them in a little freezer. So they'll freeze that okay, but to have even a small one for tissue culture, you got to be prepared to spend about $100 a month through the year constantly to keep it filled up with liquid nitrogen because the stuff boils off real fast, especially in hot weather. So that's a constant expenditure, about $1,200 a year just to keep your liquid nitrogen going. And like we said, in a few years, the cells are not going to be usable unless you take them out, thaw a few vials, and start them growing just like we started growing things from the tumor in the beginning, and let them repair their DNA and grow and get stabilized, and then you freeze them back down again. So every few years, you got to repeat this whole process, which easily runs into thousands of dollars every few years, right? For a handful of maybe a couple dozen vials of frozen cells. Okay, well, I have pretty much got to where I intended to here. So if American Type Culture Collection is charging $256 a vial for HeLa cells, do you think they're pocketing a huge profit from that? Actually, they're taking a loss. This is a cell repository. It's a service to the research community, just like the famous seed banks are to the agricultural community. But the prices that you're dealing with mean that that vial of cells, depending, and there are a lot of variables, like cost of fetal bovine serum goes up and down with the popularity and price of beef. But it is true that they are probably spending on the order of at least that much just to produce that vial of cells. Now, the other little thing I neglected to mention, you want to order some frozen cells. How are you going to get them to you? Mail them? Not going to work. Remember, they need to be fed every 2 to 3 days. Okay, that's a problem. They need to be in a CO2 incubator environment, so if they get chilled or if you screw the cap on and leave them, they'll die in a day or so. So what do? Well, you ship them frozen on dry ice. And you ship them FedEx special overnight. Regular overnight isn't good enough because they'll sit on a hot FedEx truck, and there's a problem because dry ice will explode. As any undergraduate who's ever played in a lab has found out, if you seal it up in a plastic container and just leave it in the sun, sooner rather than later, kaboom, it expands. And so it's a hazardous material. The bottom line is you'll end up paying about $100 just to ship a vial of cells on dry ice. Cells don't weigh much, but you need about a pound of dry ice to ensure that they arrive frozen. So add that into our equation and you can see, well, yeah, whatever else is going on here, George Gay and others certainly didn't get rich off of these cells. And so I would argue that whenever you are told that something is unethical for one reason, which it may or may not be, unethical reasons of obviously patients' rights, tissue donation, should somebody be able to profit by something that is profitable, fine. But then we tend to put a rider on, oh, and look, somebody's probably making money off of this, and that makes it even worse. Well, I don't really care if the person who robs my house is a successful thief and got rich by doing it, or a catastrophe of a thief and is not getting any better off. My house was still robbed, right? And so this is a kind of an argument that for basic research stuff doesn't go much of anywhere. Now, what about research? Profits from that, the famous $1,000 cells from Invitrogen, which by the way is now Thermo Fisher. If you want to look them up online, you can see this stuff. I'll stop with this. Thermo, because they make tissue culture incubators. And they've taken over the Invitrogen line. Well, if you look at what they've got, they have got cells that genetic engineering techniques have been used to modify radically from HeLa cells into what we call reporter cells that are very useful if you're trying to untangle one of these cell signaling things. If you take cell biology, I guarantee you will never think the New York subway system is complicated again, because the cell signaling pathways, when a hormone speaks to a cell on the surface, there are all kinds of reactions that happen inside that cell that are tremendously complex. What are you going to do about that? How are you going to sort it out? How do you know which of the many, many actors has been activated by phosphorylation or something and which is not, to dissect these pathways? Well, one way you can do that is to buy these engineered HeLa cells and inject a little of your cell in question into them and see if they light up, because they've been engineered with fluorescent proteins that will light up if the certain signaling molecule in question is working. And that took probably at least a year of several PhD level people's full-time efforts and reverses, and I don't want to tell you how many hundreds of thousands of dollars of tissue culture reagents and fetal calf serum and all the rest to generate those. Now, if you can make a useful product out of the cells, then retrospectively, shouldn't somebody that donated the cells be able to be enriched by it? That's another question, and a very legitimate one. But at that point, I think I've said as much as we need to give you a basic feel for the science of it. And so I think it's more than time for your questions on anything like this. Remember, I will tend to give you the science answer and not the ethics answer, although I certainly think about that. That's not my expertise. Yeah.
Is there a specific reason that we use bovine serum?
Oh, yeah. You can work around this. Some cells actually will grow without any serum. They're usually cancer cells. Remember, cancer is activated to grow without getting growth signals. And we really need, the reason we need fetal serum is not because people are evil Dr. Frankensteins, but because only in the fetus do you have all this rich mix of growth factors telling cells, grow, divide, do your thing. And so we don't have that. And cancer cells will grow in serum-free medium. Some cells, like HeLa cells, will grow in less. So sometimes you can grow them at even 5%. And there are other workarounds, but they tend to be specific to particular cells. So when you're starting, you probably have to use fetal bovine serum. Fortunately, it's fetal bovine no matter whose mammalian cells you're growing, so we don't have that issue.
As a follow-up to that question, do you think that some of our practices in tissue culture are sort of historically just practical? Like,
Oh,
we
yeah.
tried it and it worked, like the 5% CO2.
Yeah.
I mean, that's not physiological, right?
The which?
The 5% carbon dioxide.
No. If you had 5% CO2 in your blood, you'd be in bad shape. Yeah. This, by the way, something you all ought to know about science, and Ann brings this up for a very good reason, is that we're trying to get results. And if you tell people, does every step of what you're doing, including that part where you spin around and pray to the lab gods to make your experiment work, are you sure that really is necessary? An honest scientist is going to tell you, heck no, all I know is it works. And I haven't got the time to stop and investigate each and every step. And what happens is that you do that when you're forced to. So when the HeLa contamination made people realize that reusable glassware, just like reusable syringes, might be a bad idea, might lead to contaminating one culture with another. Then they started spending the money for plastic disposable ware, but not before. Nobody said, I want to do an experiment to see how frequently my cultures are cross-contaminated by using a glass pipette, because basically you can't publish that. So, yeah, so could it be improved on? Definitely. Could it be cheapened? Well, if each and every one of you takes up as a hobby growing cells in culture, and it spreads like wildfire and becomes at least as common as model railroading or stamp collecting as a hobby, chances are no, because we're talking about a very limited audience here. And when you have to produce something for a small number of people, there's usually no good way to do it very cheaply. So I don't foresee the economic improvement. But yeah, could other things be discovered as we learn more about growth factors, identify them more? No, they're very expensive right now. They could become cheaper, so you could have a fancy substitute if you know what your cells require. But of course, if it's a new culture, you don't. You have to learn that.
I have another question.
Yeah.
So one of the parts, and I haven't read the book since 2011, but one of the parts that really made me bristle was at some point, I believe someone gets a tour, I think it's the author gets a tour of a laboratory from a postdoctoral fellow.
Oh, yeah.
And he seems to explain that HeLa cells can fly through the air.
Oh, yeah, yeah, that's a famous myth. Yeah.
Am I remembering that correctly? Has I read it more recently?
It comes down to just about that. They can fly. They're
Yeah.
supernatural.
So you already mentioned bacteria don't fly
Yeah,
through
and
the air.
neither do cultured cells. In fact, if you take those cells, I remember having a distinguished cancer researcher come to visit our lab at Hopkins, and he wanted to see cells, including HeLa cells. In fact, come to think of it, I think he was looking at HeLa cells when this happened, and he was just fascinated. He'd never seen the living cells in culture before, and he's sitting at the microscope for 45 minutes watching these cells, and all of a sudden he says, oh, look, they're doing something. I think they're dividing. And I'm afraid I just sort of leaned against the doorpost and said, well, no, they're actually dying, because you've had them out of the incubator this long. They're rounding up because they're croaking. You killed them. They're pretty fragile, and even the robust HeLa cells can't survive out of the CO2 incubator for more than about 45 minutes, as I found, and they certainly can't crawl over a incubator stainless steel shelf. It just doesn't. Somebody decided to save time by using the same pipette to put the trypsin in each of several flasks. You have to realize nobody just cultures one kind of cell. They always work with several kinds, unless you don't want to get funded. Okay, there are cell lines for breast cancer, but MCF7s have been used for so many projects that breast cancer research panels routinely say, no, you got to use a variety of different breast cancer cell lines, or else your work is not reliable. So obviously they're going to have multiple cell lines in the lab. All you have to do is try to split them on the same day, cut a corner, and that's how your contamination happens. And of course, you never want to admit that. You always, they must have flown through the air. I'm a good researcher. All right. Okay, the other one I thought maybe, if you don't have any other questions, did anybody notice this sort of mysticism in the book? And this is something I have fought against, the whole attitude. In fact, if you want to get me to grind my teeth, just say, it's in our DNA. Okay? I mean, DNA is really important stuff, don't get me wrong, but it isn't destiny, and it doesn't have karma. It's just the stuff that contains the recipe for making proteins and guiding the expression of other pieces of DNA, which is really important, sure. But the extent to which you can say that if you took a tumor out of me and grew my cells, it would be a little bit of me out there, can be really taken a bit too far. So let's talk about, in fact, one point, a supposed researcher flat out says that if you fingerprinted the DNA of HeLa cells, it would match Henrietta Lax's DNA perfectly. Right, and anybody that knows modern biology goes, what? That's ridiculous. Okay, do you know how many chromosomes you have? Biology types don't answer. You know what the human chromosome number is? Anybody? Huh? Someone said what? Well, okay, so anybody? 46, right? 46 chromosomes. We used to say 48 because the very first pictures showed extra chromosomes that drifted over into the karyotype, and for about a year everybody was saying the human chromosome number is 48. We still talk about the rule of 48, which is most scientists can't see what's right in front of their noses. But 46 is the normal human chromosome. You know how many chromosomes HeLa cells have as an average?
54.
Yeah. Well, actually, I was checking with ATCC, and the line they're peddling right now, and you see I buy into this peddling, gives their modal number is 82. So there's a drift here, okay? 82 with a range from 70 to 105. And a dozen of them are chromosomes that we've never seen before. They're patched together from bits and pieces of other chromosomes. And in fact, if you look at the ATCC website, they have lots of information about exactly the bit of the long arm of chromosome 2 is spliced onto the short arm of chromosome 4 and so forth to make these M chromosomes, as they call them. So these are in no, and of course, you know, cancer arises as a disease based on changes to your genetic material, right? That's why things like tanning and x-rays and so on are causes of cancer. Yeah? So naturally, if you look at a tumor cell that was already a very invasive advanced tumor, you would, frankly, I would hate to have to do the job, given a tumor culture, to say, okay, we have samples of the normal DNA from 150 patients, tell us which patient's tumor this was. I think that would be very, very difficult. And a lot of the DNA fingerprinting very properly uses parts of the DNA that don't code for anything, as we used to say, controlling regions. And they're the ones most often broken up when chromosomes break and rejoin, like the HeLa chromosomes have done. So I wouldn't think of that as a part of me, I can tell you that.
So I think with some of these extra chromosomes, are some of these potentially adaptive to life in a petri dish? So that these things have now been evolving for, what, 60 years?
Well, but remember, not that long, because A, we couldn't afford to keep them growing constantly all that long. So they've had years in the freezer, and then they've been taken out and thawed and grown for a little while and put back. So assuming that if you're a cell, when you're in liquid nitrogen, time doesn't pass, more or less true, then from the HeLa cells point of view, they've probably only been growing for about 10 years. Just a rough guesstimate. So during that time, though, lots of changes happen. We know they happen in culture because every time you passage or split cells, some guys get left behind because they weren't done replicating all their DNA, right? And they're floaters, and you don't worry about them. Oh, I got the rest of the cells to grow. Well, how about if some of that slow replicating DNA controls something important? And so that's why whenever you do research with cells, there's a big important number you have to ask about. If anybody sells you HeLa cells, ask them, what passage number are we talking about? That is, how many times were they split? Because that number wants to be a real small number. The longer they've been subcultured, split, the more changes will have accumulated, and you're selecting in tissue culture. I found that out a very hard way many years ago in research on basic cancer cell biology. So HeLa cells were immensely useful as a basic tool to understand cancer. But basically now the utility has really dropped, except as a vehicle for these reporter genes. Maybe someday somebody will develop a diagnostic procedure that will actually be a moneymaker, but I'm here to tell you basic research doesn't make money. That's why it's called basic research. If we could patent it and have a big company to get rich off of it, the ideas were already generated by people like George Gay and thousands and thousands of other people today. So.
And since you raised the issue of mysticism and no one has raised it again, is that, would you like to speak to that about the book from the journalistic perspective, or because you're a scientist?
Yeah.
Is that how you think the journalistic, the way that it was written came about?
Yeah. Well, I think as a journalist, people tend to try to grab people's attention. The most striking thing they can. And when we talk about immortal cell lines, boy, that's a resonant term. What does that mean? It means that their telomeres are permanently long. And look that up if you don't know what that's an aging feature that your cells have built into them, and they short-circuited that so they don't get older the way your cells would if you grew them in culture. There's a whole piece of this I haven't talked about. Well, to go from that to actually saying that the person has become immortal in some sense because the tumor that killed her has been grown in culture and has long telomeres, isn't quite, at least if they were saying that about me, I sure would say, well, that's a heck of a kind of immortality. I'll pass on that, thank you very much. And so if you want to grab your reader's attention, you kind of imply that in all those little incubators and cell freezers, there's a little bit of consciousness, a little bit of the person there. And that is going way beyond anything we can say scientifically anyway, and doesn't make a whole lot of sense to me. Now, to say, should you take a person's cells and work on them without their consent, it's another issue. But is the person there in the tumor? Oh, not in any sense that you could recover. If I gave you the lab project, go and take these cells and clone a brand new Henrietta Lacks who might get to live a full and happy life, that would be beautiful. Not very feasible.
As far as Sloop's book goes, I think that this sense of the mysticism and, you know, the idea of the immortal life is so prevalent because she's not only telling the story of these cells and the cancer, but of the family and of the children and Deborah's longing for a mother that she never got to know. And there's so much that the family did not know about science. So much, so that, you know, hearing that there are cells of my mother's that are alive. I was just rereading that part today. Where is she?
That's heartbreaking, yeah. And
Yeah.
to be honest and say she's not in the cells. I'm reminded of the young woman who spent so long in a coma because her parents were so sure that when the doctors weren't around, she opened her eyes and responded to them. And at autopsy, they found out her brain had been destroyed long ago. We cling to things. And it's not always the kindest thing to, you know, substitute scientific mumbo jumbo like, well, her DNA is there. Well, actually, it's totally messed up, and it's what caused the tumor. So that's a part of her you really don't want, if it is any much of a part of her. That's a hard thing to tell people. But yeah, there's gaps in the way people are treated, gaps in the way people are educated, gaps in all kinds of things, which as a scientist, my bottom line is I wish we did a much better job of educating people and giving people opportunities and, you know, making the world a better place. And never mind all the, you know, maybe if there's a little bit of DNA left, we could think that the person is still there. And after all, you can recover Napoleon's DNA from his hair if you go to his tomb. But does that mean he's still there? I don't think so.
Do you have time to explain how the HeLa cell would survive?
Say again?
Do you have time to explain how the HeLa cells would survive in the Petri dish when they were sanitized? How did they survive that sanitation process? I don't understand that.
In which?
In the Petri dishes.
Yeah.
You said they survived.
Oh, not when they're autoclaved. No, autoclaving will kill them. It's the pipettes, the glass pipettes that you put your trypsin in. And if you have the same trypsin solution and you're putting it into 4 or 5 different flasks of different cell cultures, you may think, well, I'm just putting something in. I'm not taking anything out. So can't I use the same pipette? Well, yeah, if it touches a droplet with the HeLa cells in it, and then you immediately go to the next flask or dish, then you contaminate it. But yeah, the Petri dish, the cells are stuck to it, so they don't get out quite so easily, but mainly the disposable pipettes are what made the big difference. Lousy for the environment, but great for research.
Do you think this has changed? Like, I mean, so these days you can write down on a piece of paper somewhere that if you die, you can donate your body to science, to research. I mean, do they keep your name with your body as a journalist?
We've got to make sure, first of all, there's an organ donor form that you fill out when you renew your driver's license. And your driver's license has a marking on it to show that. And they're supposed to take cognizance of it in a hospital. Hospitals are really skittish about this, though, and if anybody with any relation to you is in there when you die and they say, oh, no, 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 I don't want her cut up or whatever, then they won't do it. And so I think there's got to be a transformation of public awareness that, you know, after all, I mean, I was a bone marrow donor. I'm in the bone marrow registry. I never got to donate just because I think it's the right thing to do. And you don't get paid for your bone marrow cells, and it's a bit of a tough procedure. But there was a young lady in Germany who nearly needed my cells, and I was all set to donate, and then it didn't work out. And so, you know, everybody should think about that. And of course, you know the reason that's given for not even doing an autopsy? And by the way, you notice the journalistically theatrical description of Henrietta Lacks's body after autopsy, which is based on what? I mean, actually, you don't leave a body in that condition during an autopsy, or after it even for a moment. But again, journalistic license, just think what they do to your body, and well, yeah, that's what an autopsy is. If you want to know, how did the cancer get to her brain, well, how do you think you find that out? You know? So, yeah, autopsies are messy, but people now very often say of a person, oh no, I don't want an autopsy, she suffered enough. I'm thinking, if anybody thought that an autopsy was going to add one iota to the person's suffering, they wouldn't suggest it. But last I checked, they have departed. So, yeah, that's the, you know, that mentality of you must never use any part of the body, you must never learn anything from the body is kind of idolatrous to my way of thinking. But anyway.
Follow up on Sarah's question. So if you have a biopsy and you have a bit of tumor removed, and they put it in paraffin, they store it, and you sign away, you know, your rights to it, do they keep a name or an identity with that data so that they could then look retrospectively at, I don't know, outcomes, look back to a genetic analysis of the tumor at time X, what happened to that person at time Y? So do they keep these tissue samples identified, at least in an anonymous?
Well, I can speak to how it's done in breast cancer because I was the scientific administrator for several breast cancer review panels. And the way they normally do it is that each case is assigned a letter number code. But then everything relevant to the medical records goes into that letter number code so that you can conceal identities. And, you know, granted, somebody could sit down if they really wanted to and say, now, who had a mastectomy on this date at this hospital and also had the following underlying condition and so on. You could probably break it, but it's designed to eliminate the identity but keep all the medical record intact, and that probably is the best way to go.
Do you think with advancing sequencing technology that you could ever, if individual A has their genome sequence, it's in a genetic database, there's a tissue sample from a tumor prior, is there enough identity in the tumor sample?
Yeah, but you'd have to preserve the sample differently. If you put it in paraffin, it's not going to be a good candidate for DNA sequencing or much else except sections. But yeah, if you adopted a procedure, it'd probably be reasonably inexpensive to preserve a bit of the sample so it could be analyzed genetically in the future. And of course, that's one way. Some of you may know part of the subtext of this is that cancer, even in one person, is not a single disease. It's thousands and thousands of different mutated cells doing very different things. And sometimes when a tumor responds and then it becomes resistant, it's, well, you killed most of the tumor, but the genetic variants that were already there survived your treatment, and that's why you got to go back or have a worse prognosis and so on. And if it were possible to genetically profile tumors in some detail, you could actually avoid some of that, maybe a lot of it. So there's a lot of interest in collecting repositories and saying patients with your genetic type of tumor historically respond best to this therapy and not to that other one. So you could make a good decision.
Are there any more questions? No? All right. Well,
Okay. Well, you're welcome.